Okay, you can start now.
Good afternoon, ladies and gentlemen. Myself, Dr. Pankaj Parmar. I am working as an assistant professor at Dairy Technology Department of Parul Institute of Technology, Parul University, Vadodara, and I will be your moderator for today's workshop. Today, we have organized a workshop on advanced methods of food preservation, which will be delivered by Dr. Manish Tiwari. Let me introduce him. Dr. Manish is a food technologist. He holds a doctorate degree in food technology from College of Food Processing Technology and Bioenergy, Anand Agriculture University, Anand, Gujarat. He completed his graduation and post-graduation in food technology from Guru Jambleswar University of Science and Technology, Hisar, Haryana. He has many achievements in his professional career. He was awarded a gold medal in MTech in food technology in the year 2018. He secured All India rank 6 in ICR SRF exam in food technology, which was held in 2018. He was awarded with ICR SRF fellowship from 2018 to 2021. He qualified ASRB NET in food technology last year. He started his industrial career in 2015, just after completion of his graduation. He served as a quality executive officer at Perfect Food Industries, SIDCUL Haridwar, Uttarakhand. Then he went for his post-graduation and doctorate. Now he is a scientific officer at the National Institute of Food Technology Entrepreneurship and Management, which is NIFTM, located at Kundli, Sonipat, Haryana. Now, without delaying much, I will hand over the session to Dr. Manish. Dr. Manish, over to you, sir.
Thank you, sir. It is always a great pleasure to be with Parul University, and good afternoon to all the participants who are there in the live session. And today's topic is extremely important for the food tech point of view, because without this, it is impossible to imagine the work or the research area of the food technology. So this topic is uh, very much important for all the food technologies and the allied sectors. So now I'm going to share my screen with you. Uh, is it visible to you all?
Hello.
Hello.
Yes, sir. It is visible.
Okay. Okay. So good afternoon to all the participants. In today's lecture, that is on the advanced method of food preservation technique, I'm going to cover food spoilage, food preservation, and the various methods of food preservation, such as physical, chemical, and novel preservation methods, along with the hurdle technology and the requirements in order to mitigate the food spoilage and to get the proper solution in the field of food preservation. So, without much, not much wastage of time, I'm going to start with the food spoilage. The food spoilage is the parallel terminology that is used by the food processing in terms of food preservation, and it is the stage at which the food is unsuitable to eat. These are depend on the several factors that affect the food quality uh, in terms of moisture, in terms of pH, in terms of the physical characterization and the structure of the food product, on the basis of the storage conditions, such as the oxygen level within the food product and outside the domain of the food product, and the temperature that are associated with the storage conditions. So as you see on the right hand side of the pic, that food, when the food spoils quickly, when it is moist, for example, milk and fruits and vegetables. When the food is at the neutral pH, they are readily affected by the microorganisms, such as especially bacteria. When we are talking about the physical structure, if we increase the surface area of the food, then it ultimately decreases the shelf life of the food product. If the food is not properly stored under the refrigeration condition, then it also affects the quality of the food product. So, what are the various types of the food spoilage? The various types, it includes the physical, that includes the mechanical handling or the mechanical disruption of the food during the transportation or inline processing. So there are a number of abbreviations and external shocks affects the surface of the food and ultimate affects the quality of the food. That's the physical quality of the food. Chemically, when the food is oxidized, the food, especially the fat-rich food, it converts into the free radicals and affects the overall quality of the food. Especially as per technical terms, you can say rancidity. There is a putrefaction, there is a lipolysis within the food. Other than this, microbiological spoilage can be caused by bacteria, yeast, and mold. And microbiological food spoilage that can be observed due to the infestation of the various insects and rodents within the food product. So how can we mitigate or how can we cure this food spoilage? We can convert it into the moist food into the dry form, or by adding certain antioxidants or buffering agents in order to lower down the pH, because certain microorganisms and enzymes, they have the optimum pH activity. They are working, their growth growing under the optimal pH. So it can be converted, if we convert or lower down the pH, many number of the microorganisms can be uh, inhibited or killed at the time. Or we can process whole meat instead of sliced meat, or we can refrigerate the meat products or food products into the for a specific period of time in order to increase the shelf life of the food product. So we can say that spoilage can be delayed or minimized by means of food processing. So when we are talking about the food processing is the multi-uni-operational measures which includes canning, fermentation, freezing, environmental packaging such as MAP, modified atmosphere and controlled atmosphere packaging, pasteurization, smoking, and added substances such as food preservatives. So, for example, if I'm taking apple, the average shelf life at the counter of the apple is 5 to 7 days. But if I can process these apples into the several food commodities by means of food processing in order to increase the value addition as well as the shelf life of the apple. So by this way, I can say that processing is also a part of the preservation. Now, this is the main terminology of the food preservation. It is the technique by which the food is physically, chemically, and nutritively safe by means of several preservation methods. When we are talking about the preservation, we are mainly focused on the microbes. But other than these, we also have to retard the oxidation of fat, inhibit the natural aging, and the retardation or minimize the discoloration. So why the food preservation is important? What are the various key elements by which we can preserve the food? So food preservation is mainly uh, done to smoothing the food supply chain process. Food supply chain from farm to fork, if there is a food preservation, the process is smooth all over the chain. Second one is it helps to add the variety uh, in your diet. For example, right now this is a season of jamun. May to June, May to July is the season of the jamun. If I converted that jamun into jamun flakes and ultimately that flakes converted into powder. So I can add something value in the jamun processing. So it adds variety also. Along with this sustainability, it maintains the food resources for the future generations, natural food resources for the future generations. Also, food preservation minimizes the processing time and energy. It also, for as I earlier said, the jamun, the availability season of the jamun is right now in the May to June month. But as I processed it, it will be also available in the winter season also. It also balance or maintain the price stability. It reduces the food wastage and increase the shelf life of many food commodities or perishable food commodities. So these all parameters raises the food value among the consumers and also boost up the confidence among the food business operators. So right now, how can we achieve the food preservation? There are multiple principles behind this. The food preservation can be preserved by means of microbial decomposition or by chemical or biochemical reaction that takes place within the food or by prevent them from the microbiological agents. So, how prevention or delay of microbial decomposition of food we can carry it out? It can be carried out by keeping out of microorganisms from the food system, by removal of microorganisms, by hindering the growth or activity of the microorganism, by killing the microorganism, and how can we minimize or cease the chemical reactions, biochemical reaction within the food by destruction or inactivate the enzymes because enzymes are majorly trigger these chemical reactions within the food system during the storage or by prevention or delay the purely chemical reaction. Prevention of from macrobiological agents such as insects, animals can be done by means of fumigation, and from the mechanical shocks prevention, it can be done by cushioning or suitable packaging material. So, when we are talking about the prevention delay of microbial decomposition, first of all, ultimate target of each food business operator and being a personal, I want to keep my food safe and isolate it from the external environment. So there are natural barriers that are provided. For example, in egg you see the cuticle layer around it, which protect it from the external environment. That is a natural barrier. Okay, but when I provide asepsis condition is what the hermetically sealed container or hermetically sealed pouches through which I can isolate my food product from the rest of the environment. So if I am fail to keeping out, if it unintentionally it comes into my food system, then my next process is to remove it. Remove it by means of filtration, centrifugation, sedimentation, trimming or washing. Trimming or washing we are using in the fruits or vegetables, whereas centrifugation such as bacterifugation we are using in the milk or milk processing industry in order to remove the microorganisms from the milk. So if I'm failed here once again, then my next step is to hinder or inhibit them by keeping them at the low temperature, drying it, provide anaerobic conditions, or treat it with the preservative so that I can able to hinder the food product from the microorganisms. If all the three were failed, then my final step is to kill them, and killing by means of boiling, pasteurization, sterilization, and radiation. Then prevention or delay of cell decomposition of food that can be achieved either by destructing or inactivation of food enzymes by means of blanching or boiling, because every food enzyme or the enzyme, they have the specific temperature through which they can grow or they activate, they initiate the chemical reaction, but at certain temperature, they inhibit, and they are inactivated. So by means of blanching, and which can be carried out in the vegetable, fruits and vegetables, so we can minimize the uh, enzyme level within the food system. In chemical reactions, we can add antioxidant so that the free radical oxygen, they can attach and remain and make the oxygen unavailable for the microorganisms or biochemical or the reactions, so that it minimizes the chemical reaction. The third one is the prevention of damage by the insects, animals, or mechanical causes. So these are some of the cushioning materials. You can able to see this how the foods are segregated from each other by means of cushioning material, and where there are proper packaging so that it absorbs the external mechanical shock during the transportation and handling of the food product. So there are various methods of food preservation by which we can preserve the food. It can be preserved by means of physical, chemical, and biological means. In physical processing, there are thermal and non-thermal processing. In thermal, there are various processing techniques that are pasteurization technique, retorting, aseptic processing, semi-aseptic processing, cold treatment, drying. Whereas in non-thermal, irradiation, ultraviolet radiation. Uh, in chemical processing, there are usage of several chemical preservatives or pH controlling agents, and by means of biochemical, we can preserve the food by means of fermentation. So other than these, there are novel preservation methods are also there. So we will discuss it later. So first of all, physical preservation method. The physical preservation method first is pasteurization. As we all familiar with the terminology, pasteurization, in which the food is partially sterilized below the temperature of 100 degrees centigrade. The birth of this process was named after the French microbiologist Louis Pasteur, when in 1860 he researched and revealed that thermal processing would deactivate unwanted microorganisms in wine. So from there to now, the pasteurization is widely used and considered as an important unit operation in wine, beer, fruits and vegetable industry, dairy and dairy processing sector, in the cheese, whipping cream, and fruits and vegetable processing industry, and the egg processing industry. So the pasteurization process is normally carried out by the industries in three ways, that is LT-HT, low temperature, high temperature, short time, and ultra high temperature. So in the case of milk, LT-HT is a batch type process in which the milk is pasteurized at 65 degrees centigrade for 30 minutes in order to remove or kill the vegetative pathogen. Whereas STST process is a continuous process in which the milk is treated at 72 degrees centigrade for 15 to 30 seconds in order to kill vegetative pathogens. While ultra high temperature is a continuous process in which milk is treated more than 135 degrees centigrade for a few seconds in order to kill all the bacteria and the spores. The shelf life of the milk of LTST process is 1 to 2 days to several days. For STST, from 2 to 3 weeks when refrigerated, and ultra high temperature for 6 to 9 months when aseptically packed. So here you can see there is a time temperature combination. As the temperature increases, the processing time decreases from 30 minutes to 15 to few seconds. So how does the industrial operators, they are using these time temperature combination. So pasteurization is not only carried out in the milk products, but it also carried out in various food products in order to inactivate their target uh, applications. For example, in fruit juice, the main purpose of the pasteurization is to inactivate the enzymes that are pectin esterase and polygalacturonase enzymes, in which the fruit juice is pasteurized at 77 degrees centigrade for 1 minute. These enzymes are mainly responsible for the thickening and sedimentation of the fruit juice. So in order to remove these enzymes, the pasteurization is generally carried out in the fruit juice industry. In beer processing, the main purpose of the pasteurization is to destruct or to remove the spoilage causing microorganisms like wild yeast and lactobacillus species. In this, the beer is processed under the temperature of 70 to 74 degrees centigrade for 1 to 4 minutes. In the case of milk, the index microorganisms for the pasteurization is Coxella burnetii, which is mainly responsible for the Q fever, and Mycobacterium tuberculosis, that is responsible for the TB, which inactivated or eliminated from the food milk system at the temperature of 1.5 degrees centigrade at 15 seconds. It is also used in the egg processing industry in order to destruct the egg index microorganism, that is Salmonella Stephenberg, at 64.4 degrees centigrade for 2.5 minutes. So when we are talking about pasteurization, the similar terminology, that is sterilization, is also comes into the mind. So here is the difference between the pasteurization and sterilization. On the basis of temperature, pasteurization is the mild heat treatment, or you can say partially sterilized treatment, in which the food is treated below the 100 degrees centigrade, whereas in sterilization, the treatment is carried out more than 100 degrees centigrade. In pasteurization, the main target is to remove pathogens, whereas some viruses and spores of microorganisms remain live in the pasteurized food. Whereas in sterilization, bacterial species, spores, and thermophiles, they will remain, but vegetative spores, they can be eliminated from the food system. In pasteurization, there is negligible changes in the nutritional quality of the food profile takes place, whereas in sterilization, as the temperature is quite high, more than 100 degrees centigrade, there is a gradual denaturation of proteins there. It affects the fat and sugar also, and also decomposes some of the calcium and minerals and vitamins also from the food system. The pasteurization is normally carried out for the acidic food products, because acidic food products, they have natural antioxidants, and they are mainly spoiled by the yeast and mold. So yeast and mold require less temperature as compared to bacteria. So that's why for the pH less than 3.5 for the acidic food, normally pasteurization is carried out. But for the sterilization, as we earlier read about this, that neutral pH are dominant factor for the bacterial species growth. So for greater than 4.6, we have to sterilize the food. For example, vegetables and meat and meat products. These can be sterilized, or these can be retort under the canning. So the shelf life of the pasteurized food products are from few days to a week, whereas sterilized food products, they are kept for several months without and under refrigeration condition. So here is the one of the product that available in the market, that is sterilized low fat cream. So here you can see it is packed into a can. So there is a terminology, that is canning, because sterilization is carried out more than 100 degrees centigrade. So there is a process of canning in which the cans are um, the cans are kept under the cans are processed under the retort conditions in order to achieve the commercial sterilization. So what is commercial sterilization? The commercial sterilization is probability a probability that out of how many of cans, say if I'm treating 100 cans, if I'm retorting 100 cans, the probability of the can that one can out of 100 is faulty. 100 cans, only 99 cans they are microbial free, and one can maybe a chance that it will might be a condition that it will contain microorganisms. So that much safety level we can achieve as per regulatory condition. Now another preservation technique is the chilling or the cold storage. The chilling is mainly done to retard the changes or inhibit the time lapse of the microbial growth or the chemical or biochemical changes. For every 10 degree centigrade fall in temperature, two-fold decrease in rate of reaction takes place. That by this, the by this the chilling process is used to extend the shelf life and freshness of the food commodities. It also causes minimal changes into the sensory characters as the process is carried out under low temperature. So there are minimal changes in the sensory characteristics and the nutritional properties. The chilled foods are perceived by the consumer as being convenient, easy to prepare, high quality and healthy, natural and fresh. These are used not singly, these are used with the other unit operation also. For example, pasteurization, because after pasteurization, immediately we have to cool the milk to 4 degrees centigrade, and sauce wise, where the vacuum packed food products are cooked for within a range of 50 to 70 degrees centigrade, followed by chilling. So chilling is not a single mode factor. It adds with the other unit operations also. Chilling also combined with the control of the composition of the storage atmosphere, such as modified atmosphere storage or controlled atmosphere storage. So here there is a storage technique that is zero energy cooling chamber. As the name suggests, it includes it consumes zero energy, and it can be easily manufactured at the ground level, where the hot, arid or arid climates are there, and there is an electricity crisis. So this type of how it works. It works on the basis of evaporative cooling. By which the small scale farmers, they are able, who they are not able uh, to galvanize the proper warehouses. So in order to reduce this is the cheapest and most economical technique of the cold storage. Then how it is work? If I can say, for example, uh, these are the construction unit of this zero energy cooling chamber. In this, the uh, the cavity is filled with the sand, moist sand within the two walls. Uh, there is a dig between these two walls. In these, potatoes are kept under the specific conditions. Uh, when the potato releases the heat, the released heat from the brick, it will pass through the moist sand, and moist sand is continuously sprinkled water on the sand, and this moist sand releases heat from the in the form of vapors outside from the system. So that way the interior system of the zero energy cooling chamber uh, works and increase the shelf life of the food. So these are some of the conditions on which the fruits and vegetable storage conditions can be carried out under the storage temperature relative humidity that increases. If these are not proper, then it diminishes the shelf life of the food product. So next one is the freezing. If we are talking about the freezing, the food is going with the different phases when we kept the food below the 0 degree centigrade temperature. So in the freezing, there are different phases occurs from A to F, as we can see here. From A to B, there is a super cooling stage. As at this time, the water remains in liquid due to the presence of solutes present in the food system. So it will not uh, exactly freeze at 0 degree centigrade. It lowers down, it depresses from the 0 degree centigrade and converted into food product. Then the temperature increases as the supercooling stage. The product starts nucleation. At the B point, the product starts nucleation within the system. In this nucleation, what happens when nucleation is the formation of ice crystals. During the formation of ice crystals, what happens? The release of heat taken place. So when release of heat taken place, what happens? The temperature increases to some point. At C point, B to C, there is a crystal growth stage. Ice crystal begins to form, and in the C or D, C to D, it is continuously formation of iron crystal around the nucleation, ice crystal around the nucleation. So as the continuous crystal formation, the temperature uh, again goes lower down. When there is complete formation of ice crystals at D point, then there is a release of heat from the solid system, a solid ice crystal system. So there is a elevation deviation from the system taken place. So from here, the ice forms, and at E, E point is the thermal arrest time stage. D to E, thermal arrest time stage, from where all the heat uh, cease or arrest at this point, which is known as the thermal arrest time point. Followed by this, there is solid cooling takes place, which is further gradual of removal of temperature from the system. So, why the thermal arrest time is more important? Because it tells you about the whether your process is slow or fast freezing. If it is slow, you can say the big tail of this. So if it is slow freezing, then it will take more time to frozen that product, and there are more nucleation, more ice crystal formation takes place that results in the formation of large ice crystals and ultimately affects the product texture during the whole process. Whereas in fast freezing, there are formation of small ice crystals, immediate and small ice crystals. So if the total time, if you are consider if the time is freezing time is more than 30 minutes, if this thermal arrest time is more than 30 minutes, then we can say that the process is slow. And if the temperature is less than 30 degree centigrade, 30 minutes, then we can say that the process is fast, and the fast freezing gives you the best quality product because of the formation of small ice crystals, and therefore it maintains the texture of the food product. Another preservation technique is drying. The drying is mainly uh, uh, deal with the moisture content present within the food product or the water available within the food product. There are three modes of water, modes of water that are present in the food system. That is free form of water, bound form of water, and water of hydration. So in this picture, you are clearly able to see these are the solid parts which are surrounded with the bound water along with the solute, and these blue color, it shows the free water. So while we are going with the drying, so first the free water oozing out from the food system. That's confirmed up to a certain level. So you are able to understand that no food has no dry food has the moisture content of 0% moisture. That is because of this. So when we are talking about the moisture, free moisture, bound moisture, and all, we are talking also about the moisture content and the water activity. So what is the difference between these two? Moisture content is the amount of total water, that is bound water and free water in a sample. It is a quantitative measure of the water present in this sample. That how much water is present. It will give you a quantitative information. But in water activity, it tells you about the free water or the available water that is present in the food system. Microorganisms, what the microorganisms require? They require easy medium to uh, grow, to feed. So for them, easy medium is free water. Okay. So free water, they are easily available for the microorganism. If we reduce this free water or if we calculate the water activity, it will tell you that the product is stable for a longer period or not. So for this, there are various water activity levels for certain microorganisms such as bacteria. For bacteria, it is 0.91. For yeast, 0.85. For mold, it's 0.80. Halophilic bacteria, 0.75. Xerophilic bacteria for 0.65. Osmophilic bacteria, water activity is 0.60. Below this 0.60, we can say that our product is microbial free. But for the commercial products, the products that are available in the market, they set the value of 0.30 to 40 of the water activity, so they can achieve the self-stable product within the market. So there are various factors during the drying that are taking place that include initial moisture content of the food product. If initial moisture content is high, then it will take more time to dry. Then chemical composition of the raw material. If it contains more fiber or more carbohydrates, it requires more time to penetrate the free form of moisture to the surrounding. Then shape and size. If the product have even shape, then the drying rate is faster as compared to the uneven shape or size product. If the temperature is high, then rate of flow of interior moisture to the external is higher. If the humidity is low in the atmosphere, the rate of transfer of moisture is higher in case of lower humidity. Air velocity, as the air velocity is increases, the rate of drying rate also increases. Pressure, for example, vacuum drying under reduced pressure, the boiling point of water immediately achieved, and it reduces the drying time of the food product. Then heat transfer, the heat transfer in the type of heat transfer that is in the form of conduction, convection of radiation. So it depends on all those factors to achieve the proper drying. So these are some of the novel drying techniques that comes under the novel drying techniques that comes under the consideration right nowadays. These are natural modes of drying, which include greenhouse drying, evacuated tube heat, pipe solar drying. Whereas in mechanical mode of drying, there are vacuum drying, refractive window drying, infrared drying, microwave assisted drying, fluidized bed drying, spray drying, and freeze drying. But in these techniques, there are two modes of drying. We ultimately have the aim to reduce the moisture. We do not have to what you can say that you have to elevate the temperature. In freeze drying, under sub-zero condition, we also achieve the same drying uh, phenomena in order to remove the moisture from the food system. But what is the difference between these two? here you can clearly visible from the image that the water and the solid and the flavor compounds, they are free. They are free and they diminish from the food as the air drying occurs. There is no entrapment. But in freeze drying, there are the ice crystals in which the flavor compounds entrap within the ice crystal. So it maintains this. The pores left by the ice circle, this initiates the drying and helps to retention the flavor component. So this is the major difference between the air drying and freeze drying. That's why the freeze drying products, the freeze dried products, they are uh, nutritionally nutritionally superior, and they have the more retention of phenolics and bioactive components. Another technique is irradiation technique. Irradiation technique in which the food is treated under the radiation. It is also known as cold pasteurization technique. In which, why? Because the food is preserved and the microbes are killed under low heat or no heat. So the irradiation can be achieved either by beta beams of radiation, gamma beams, or X-rays. But commercially, gamma radiations are normally used in order to achieve the cold pasteurization. So how this can achieve at the commercial level? It depends on the principle that it directly destructs the cell membrane of the uh, microorganism. So directly. So it directly penetrate within the cell membrane and affects the DNA, the dimers that are present within these DNA. So it directly affects and destructure them and ultimately cause the cell death. Whereas indirectly, how it happens? It forms oxygen radicals that penetrate onto the cytoplasmic membrane uh, and seize their activity from there, and ultimately that inhibition of phosphoglycodins on the cell membrane, it will cause the DNA damage that will cause the cell damage of the microorganism. So it is a multitasking process and can be used for several reasons. This can be used in order to route inhibition for the onion, tomato, potato, ginger, garlic, for insect disinfection, cereals, pulses, and dry fruits. It can be used to increase the shelf life of the products such as chicken, meat, and fish. It is also used for the pathogen reduction in the spices or flesh of the foods, and also quarantine the foods in order to delay their ripening process. It depends on the various doses. Um, so how can irradiation of food can be processed? For example, if I'm taking dried stuff of the moisture content 14%, for example, cereal grains, it has the moisture content of 14%, followed by sorting, clean, and grading. Then there is a test called bioburden contamination test, in which the initial contamination level of the microbes within the food samples can be estimated before the sterilization. After this, the product is primarily packed, then secondarily packed, then sealed it, then treatment was done. Treatment can be done in the irradiation chamber. Then the treatment is saved and the observations are recorded. Then the the evaluation done in the laboratory. If it is within the safe limit, then it will proceed to the market. So that is how the process of the irradiation food takes place. So these are some of the regulatory limits from which the and can be divided on the basis of their dose. Up to 1 kilo gray, it is considered as the low dose, whereas 1 to 10 kilo gray, it is considered as the medium dose, and high dose is considered under 10 to 50 kilo gray. So So in low dose, what we can able to inhibit the sprouts, insect disinfections and parasite disinfection, delay of physiological processes such as ripening. Whereas in medium dose, it is used to extend the shelf life of the food product. It eliminates the spoilage causing pathogens and microorganisms, and it also affects. It also removes those that affect the food properties, such as if I want to increase the yield, then irradiation technique, because it ruptures the cell and it releases the juices from the interior cell product. So for this, it's also used within the range of 1 to 10 kilograms. For high dose, it is used for the sterilization in the hospitals or in the perishable food commodities. Whereas it is also used in the decontamination of the certain food additives and ingredients, such as in the case of spices, enzyme preparation, natural gums, etc. So if the product is irradiated and supplied to the market, then there is a logo. This is the logo that is known as the Redura. This logo, as per FSSAI, it is mandatory to impose that logo on the food label. So another preservation technique is the chemical preservation, in which under 3.1.4 FSSAI, the preservatives can be classified into two categories: class 1 and class 2 preservatives. Now, what are preservatives? Preservatives are the substances that are helped to preserve the food. So these can be common salt, sugar, dextrose, glucose syrup, spices, vinegar, honey, edible vegetable oil. So these are the chemical preservatives that are naturally you are observed in your kitchen level, and these are naturally processed. So these preservatives, they do not have any restrictions within the food system. You can add as per your requirement. But for class 2 preservatives, these are the synthesized preservatives, such as benzoic salts of acid, salts of sulfurous acid, nitrates and nitrites, sorbic acid, propionate, lactic acid, acid calcium phosphate, niacin, propionic acid, and sodium diacetate. So these for class 2 preservatives, the use of more than one preservative is prohibited in the case of class 2 preservative. So when I'm talking about the class 1 preservatives, you all know about the salt, as it is used to preserve or cure meat and several vegetables, because these two have the pH near to the neutral. So they are readily affected by the bacteria. So in order to cease their activity, microbial activity, uh, salt can be used as a preservative, and how it can work? It works on the basis of osmosis, from which the salt molecules penetrate within the food product, and the water molecules ooze out from the food product, by which the decrease in the water content within the food product. As you see, the pickle industry, you have you think about that the mango pickle industry, how they process pickles in winter, because because mango season of mango is right now, is the summer season, but how they can achieve? They can achieve by this, by dipping that mangoes into the pickle, into the brine solution. So so they can use it in the off season also. And then second class 1 preservative is sugar, and several products we are getting into the market in the form of jam, jelly, murabba, leathers, candies, etc. So these all are the sugar-based class 1 preservatives. Next preservatives are spices that can be used as whole, as ground, or their extract can be used to preserve the food. So here is the example of the pickle, in which you are clearly see the ingredients that includes all the class 1 preservatives. That is the main ingredient: mango, salt, corn oil, chili, mustard, fennel, fenugreek, turmeric, asafoetida, and acidity regulator, that is acetic acid E260. So you can see that all the class 1 preservative are associated except sugar into this. Next class 1 preservative is dextrose. As dextrose is not as much sweeter than the glucose or sucrose, but its glycemic index is quite high, and it is around 100. That means it readily provide energy to the body when you consume this. So with this application, several food industries uses in wine or in ice cream mix or sauces or wafers or fruit cakes and ready to cook chapatis, uh, because it because it provide not only provide the color, but with the low calorie, sorry, with the high calorie, it also provides the quality color to the product. So that's why it is extremely used in the food industry. Class 2 preservative includes the acetic acid, which we can obtain from the natural source: vinegar, benzoic acid. They are naturally present in cranberries, plums, prunes, cinnamon, cloves, and most of the berries. They are associated with these benzoic acids. Lactic acid, that are obtained from the lactic acid bacteria. Propionic acid, that are obtained from the Swiss cheese, mainly by using Propionibacterium prudentii, and sorbic acid, that are obtained naturally from the round berries. So class 2 preservatives, they are benzoic acids. They are mainly they are mainly used in the acidic foods like fruit juice, jams, and pickles in order to inhibit or kill the microorganisms such as yeast, mainly yeast followed by mold and bacteria. They effectively work at the pH within the range of 2.5 to 4. It inhibits the cellular uptake of the substrate of molecules, and because of this absence of the substrate, they are not able to get the substrate. Then microorganisms, they are not able to grow. So the permissible limit within the product is 0.10%. Propionate, as you know, several propionates are used in bakery products or cheese products, in order to inhibit the mold, yeast, and bacteria. It effectively works below the pH of 5.5, and how it affects, it inhibits the microbial growth by competing with the alanine. So whatever the dimers they have in the DNA, it affects the alanine or other amino acids which are needed for the microbial growth. So permissible limit for the propionates in the food product is 0.32%. Then sorbic acid. Sorbic acid, again, it is used in the acidic foods, cocktails, dried food, cheese, pickles, baked goods, in order to inhibit mold and yeast. Uh, uh, it effectively works within the pH range of 4 to 6. It affects the membrane transport and cause subsequent cell death. If the permissible limit of sorbic acid is less than 0.20%. The nitrites and nitrates, as we all know, it's used in the meat as a meat curing agent and used in the meat and meat products in order to inhibit the Clostridium botulinum. It prevents the ATP synthesis that convert into pyruvate. So it sees their functionality and ultimately ultimately inhibit their growth. So the maximum permissible limit is 120 ppm for the meat product. When we are talking about the benzoic acid, uh, it is effective uh, under undissociated form. Undissociated means it will not readily soluble in water. If it is soluble in water, it is known as dissociated. So it is not, it doesn't show as much antimicrobial effect. It reduces the pH of the solution, not uh, uh, inhibit the microorganisms or microbial activity. So if we minimize uh, the microbial activity, we can use it in the undissociated form. Then sulfurous acid or the salt, they can use to retain the fruit color in the form of SO2. They are also used as a bleaching agent in the sugar processing industry. They are also used as a color fixation in minced meat, as they as it maintains the uh, heme iron in the reduced state and prevent from the gray discoloration. It also used as a whitening agent in the form of potassium metabisulfite in mushroom processing. In wine processing, it inhibits the growth of the or kills the bacterial growth. It prevents the wine from the browning and oxidation and certain substances that can be reduced, and it also improves the taste of the wine. So several food commodities in which the sulfurous acid can be used in ppm. The cider or wine, it can be used up to 70 ppm. Beer or malt liquor up to 15 ppm. Fruit juice, jam, jelly, tomato ketchup, and mincemeat up to 500 ppm in the form of SO2, and dried fruits and vegetables as 2500 ppm as SO2. Another class 2 preservative is sodium diacetate. Uh, that is a mixture of sodium acetate and the acetic acid. It is also known as diacetic acid. It acts as a fungicide and a bactericide within the food products such as cheese, fat, butter, bread, meat, and poultry products. The permissible limit of the sodium diacetate is less than 0.25%. Another chemical preservative is the acid calcium phosphate, that is the acidity regulator and maintains the pH within the food system. It is used in the baking powder and acts as a bread enhancer, or you can say raising agent, as it strengthens the gluten properties within the bread. Whereas it binds the metal ions, as it is this is a calcium ions. It is behave like a chelating agent and binds the metal ions and increases the antioxidant activity within the food system. It stabilizes the texture of the canned vegetables. Another class taking preservative, or you can say it's a biopreservative, that is lysine. Lysine is a 34 amino acid peptide that is mainly obtained from lactic acid bacteria. It is effective within the pH of 3.5 to 8. It uh, kills, or it shows the action against the gram-positive bacteria and their spores. The dose of 1 to 25 ppm is required to kill the microorganisms, specifically gram-positive bacteria. Commercially, it is naturally present within the dairy products, but it is present in less than 1 ppm. So it can be additionally added from the external source by using lysine, which coded with the E number of 234. So how it kills or inhibits the activity of the microorganisms. So you can see the lysine molecule. It it binds within the cell membrane and produces the and formation of pore has been done. So with this pore, there is inhibition of peptidoglycan synthesis. By this way, it minimizes or it inhibits the microbial growth within the food system. So these are some of the products in which the niacin is used. These are processed cheese, uh, pasteurized milk and milk products, pasteurized chilled soup, crumbs, canned foods, ricotta cheese, and continental type of cooked sausages, dipping sauce, salad dressings, beers, and processed meat with the specific level within the range of 1 to 25 ppm for the above mentioned food product. For the processed cheese, it is 200 international unit per gram of the level of niacin used to kill the target microorganism. So here we are talking about the physical and chemical preservation method, but there are several limitations which are associated with this, that they alter the natural taste, flavor, texture, and nutritional properties or medicinal properties of the food. So in order to avoid them, there is a uh, birth of novel preservation method uh, that includes ultraviolet radiation. Ultraviolet radiation is an radiation technique other than irradiation in which the food is processed within the range of 400 to 100 to 400 nanometer wavelength of light source. Uh, in ultraviolet radiation, the UV radiation categorized into three categories: UVA, UVB, and UVC, with this range of wavelength. Uh, so for UVA, it is 315 to 400 nanometer, whereas for UVB, it is 280 to 350, and for UVC, it is 100 to 280 nanometer uh, wavelength of ultraviolet radiation used. Among these three, UVC is effectively used against the microorganisms inhibition, and it was researched and revealed that maximum efficiency of the ultraviolet was found at the wavelength of 254 nanometers. Now, how it works? Again, this is the normal DNA of the microorganisms, and this is the UV inactivated DNA. So you are clearly able to see that the thymine dimer and the adenine dimer. You can clearly, these are parallelly switched with each other. When UV radiation treatment is given to the food product, the microbe present within the food system, the DNA of the microbe of thymine and adenine dimer readily affected. If it affected, it loses the activity and ultimately causes the cell death. So UVC penetrates in this DNA and destroying the adenine and thymine bonds. That's the main reason that and that's the way the microbes are able to kill. So these are some of the application path or broad view into a single pic that the UV protected UV treated food have minimal effect on the nutritional properties of the food. It has less operation cost. It is a green technology as it utilizes less energy and less time, and it is highly efficient as compared to the uh, chemical and physical preservatives or ancient method of food preservative. It also considered as the clean label as it doesn't require any food additives to be a part of their food label. Along with other techniques, if we are using other techniques, it is highly effective um, for the killing of microorganisms. Another novel processing technique is the high pressure processing technique, in which the food is processed under higher pressure. It is also known as pasteurization, high hydrostatic pressure processing, or ultra high pressure processing, in which the food is processed within the range of 100 to 1000 megapascal. The components of the high pressure processing unit is called piston, vessel valve, heating and cooling system medium, food sample intensifier that uh, intensify the low pressure to high pressure, and high pressure medium tank, which includes the water. So how the process can be carried out under high pressure processing? In this, there is a basket of high pressure processing unit, in which the packed bottles or the packed food samples can be inserted, which uh, which in which the product is loaded to the system. Uh, then the system is pre-filled with the pressure medium. That pressure medium is water, followed by pressurizing. Here, the pressure medium has been filled with at the low pressure. whereas the holding pressure time, where the process is carried out under the high pressure, and then decompressor or deprocessing, where the again pressure lowers down the uh, uh, desired pressure. So how the process come up? The pressure initially when we are unloading it, there is a come up time. Come up time in order to reach from 1.1 megapascal to the desired say 100 megapascal. So from zero point 0.1 to 100 megapascal, it is come up time, the time that is required to reach from 1.1 to 100 megapascal. then the uh, then the product is hold for certain period of time, that is known as the holding time or the process time. This is known as the process time in which the product is carried out or hold for a specific period of time at a constant pressure. Then the decompression time was there in order to depressurize the product from the chamber. So as there is a linear relationship between the pressure and temperature, as the pressure increases, the temperature also increases along with time. So for the pressure, it is come up time pressure, and for temperature, it is known as the come up time pressure. And similarly for the pressure holding time and for the decompression time. So during processing of with the high pressure, two major principles are carried out there, and that is Lee-Chaklias principle and isotactic principle. When the food is kept under the process, there is a gradual decrease in the volume. Gradual decrease in the volume when we apply the pressure. It shrinks the food volume, but not but it will regain when the pressure is deployed from the system. So it's based on the Lee-Chaklias principle. And what is isotactic? Uniformly pressure acts equally in all the directions. This is the food system within the chamber, and equally all the pressures constitutes to the food system in order to achieve the high pressure process. So how it kills the microorganisms? The similar way, at the higher pressure, it increases the permeability of the cell membrane, due to which the inner material within the cell pulls out from the cell under the high constant pressure, by which there is a leaching and possible conformational changes in cytoplasmic proteins or ribosomes and nucleic acid takes place, by which there is sudden death of the microorganism. So how HPP preserves the food? This is the main requirement of the HPP, that it not only kills the microbes but also reduce the enzymatic activity. Also, it doesn't affect the covalent bonds of the food product. It only affects the non-covalent bonds. That's the way it maintains the textural properties of the food. Whereas under reduced pressure, there are possibility of reversible changes, but under higher pressure, the chances of irreversible changes take place, by which the food remains as it is created at the loading time. So it maintains the overall morphology, or you can say the shape of the food product. So now we are discussing about the various applications of the high pressure process. It is not only limited with some of the products. It is a versatile processing technique in order to preserve the various foods such as vegetable products, meat products, tea and fish, juice and beverages, and other products. Dominantly, it is being used for the neutral pH food commodities such as vegetable products and meat products. So some of the products outside the India, we are able to see some of the products that are labeled with these type of logo, that is high pressure certified. That indicates that the brand is protection against the recall. you can say you are able to see these products. Here, the logo is clearly mentioned that the product is high pressure process. So that logo indicates that the brand is protection against the recall, because the recall from the presence of microorganisms or spoilage causing microorganisms. Then it also reveals that the pathogens are in control phase and they are not affect your health. It also ensures that the minimal processing of food can be taken without heat. There is no heat treatment takes place. The preservation of uh, food quality remains as fresh as it does not alter the covalent bond. It accesses the open opportunities to the new market for the non-thermal techniques, and it also indicates the process is cleanly labeled, that that their product is not containing any food additive in there as an ingredient. So next process is ozonization technique, in which the oxygen is converted into ozone by means of higher voltage generator. How it can achieve, you can able to see at the right hand side of the image. Here, oxygen molecules under the corona discharge splits into oxygen and into the single atomic oxygen. That single atomic oxygen again combined with the O2 and generated ozone. That ozone, that single atomic oxygen disconnects from the ozone and performs oxidation and decomposition the functions, and the ozone, that molecules again turn into oxygen molecules, and this cycle is repeated continuously. So from here, the oxygen is converted into ozone, and that ozone is sent to the solvent medium that has the optimum pH and temperature, under which the foods have been sterilized and microbial free. So how it acts? It oxidizes the surface of the cell membrane. The ozone molecules, it entrap on the surface of the contaminant and uh, lyses their membrane, and ultimately cause the cell death. So these are some of the case studies that for the strawberries that are kept for 8 days. If it is ozonated water, if it is tap water, then what is the condition, as you are able to see. For brucellosis and brucellosis, these ozonated water for 16 days, you can able to see, you are able to see this. These are remaining as fresh as, but with the tap water washing, you are able to see the clear difference between these. So there is a broad application of the ozone, and its applications are mainly associated with fruits and vegetables. It is also used in the water treatment, action on pests, food packaging, spices processing, pesticide removal, meat processing, and grain processing. Another very much popular technique, non-thermal technique or novel preservation technique, is the pulse reactive field, in which the microbes are eliminated or killed by means of electroporation technique. In this, high polarization pulses are generated within the range of 10 to 80 kilowatts for a millisecond, can be are used, and by which there is alteration in the ionic charges. They are trying to, as opposite charge attract each other, these form the pores within the cell membrane, as you are able to see as the pulse generate increases. With the continuous frequency of the pulse generation, you can see the clear lysing of the cell membrane, and that destructs the cell and oozing out the internal elements from the cell there, and ultimately block the functioning of the microorganisms. So how it can be achieved within the when we are talking about the pulse electric field, right nowadays these are available in both for both the commodities, for solid, for liquid also. Um, if we are talking about the continuous flow, then for the liquid products, um, it depends on the type of the electrodes that we are using, under which the food can be supplied within the system. So you can say these are the parallel plate configuration of the electrode from which the process has been carried out. Second one is the coaxial configuration from which the pulse generation occurs from the center to the electrode to the outside, and product flow within these. And the third one is the collinear, in which the uh, there are you can able to see the black patches on this. From this, the pulse generation takes place from uh, positive to negative charge, and within this, the product is flow and sterilized. So by this, the process is intended to operate at the ambient condition. It may cause rise in temperature in the product, and it mainly depends on the field strength. Not a major difference within the temperature. No? As there is a very short form of pulse for a millisecond takes place. That's why there is no drastic change in the temperature. So, but if it goes, if it arises, you feel that there is a change in temperature, increase in temperature. So it might be due to the field strength, pulse frequency, or the number of pulses. That's why these are the input variables are very important before you carry out the experiment. So pulse electric field is a multitasking property in which it directly or indirectly influences the cells of the microbes or plant tissue. So directly, if we are talking about, it can be used for the, if the product is pulse electrified, then the redistribution of moisture, or you can say rehydration of the food product, it is better in as compared to the non-pulse electric phase. Whereas the textural properties, it has smooth textural properties with the uh, with the optimum osmotic flow of the solvents within the from their cells uh, to the surrounding. Indirectly, it is also used as a pre-treatment technique for the drying, as pulse electric field is used to accelerate the drying process. It is also used to carry out to increase the juice yield from various sugarcane juices and you can say grape juices. Or it is also used to isolate the bioactive components from the cells or the plant tissue. So there are with the terminology, if you can say applications, there are broad applications of the pulse electric field in the field of fruits and vegetables, frying, beverage, meat process, milk industry. One of the case study I want to share with you that this is the frying of potato fries. The fat content of the potato samples after frying and different pre-treatments are observed in untreated blanched or pulse electric field treated, and you can able to uh, see the change in the oil absorption. So uh, with this report, it can be revealed that the reduced fry breakage in the French fries has been observed in pulse electric field, as because of the electroporation, microporation, it increases the elasticity of the French fries. What is the main limitation of the French fries earlier, which is associated with the untreated and the blanched one, is that uh, they are very their texture is very tight and they are very hard. So while carrying it, they are uh, split easily. So they are not readily uh, used or can be accepted by the consumer. So in order to remove that some of the hotels and they are using pulse electric field in order to reduce the fry breakage during the French fries. It also adds the sustainability, or it is also considered as the green technology as it minimizes the time and temperature and the energy. And it also initiates or increases the blanching, drying, and pre-frying times, or it reduces the fat content up to 50% that you can able to see. In untreated, it is 12.13, which is reduces almost 50%, 7.44. So next non-thermal and novel technique is the ultrasonication technique, in which the cavitation results in the killing of microorganisms. The ultrasonication is the processing technique that is used on the basis of cavitation within the frequency of 20 kilohertz to 10 megahertz. There are two types of frequency ultrasound are being used. That is the high frequency ultrasound that ranges from 20 to 100 kilohertz. These are known as the power ultrasound, which is mainly used for the cavitation and mainly used for the sterilizing the food. Whereas low frequency ultrasound is greater than 20 kilohertz and it normally used for the identification of the cell cavities within the food product or non-destruction technique that it can be used. So how it principally behaves with the food system, how the cavitation occurs within the system, as you're able to see the probe within the beaker in which the food sample is carried out. During ultrasonication, there are various waves of compression and rarefaction takes place. With the continuous compression and refraction of the molecules, the bubbles formation takes place, and these bubbles are successively increases as the uh, processing as the waves goes on. And ultimately, there will be a critical size above which they are not able to bear the waves, and ultimately the bubbles impulsion takes place at 2000 to 5000 Kelvin at 300 to 1200 bar pressure. So bubble, this impulsion of bubbles removes this impulsion of bubbles creates the
Pankaj
porosity.
sir, sorry to interrupt. Hello,
Hello.
Pankaj sir. Hello, Pankaj sir.
Hello. Yes, sir.
Sir, we are having another session at 4 p.m.
Okay, sir.
Huh.
So we need to wind it up. Uh, hello, Manish sir.
Hello.
Hello. Yes, sir. We have a parallel session. So
Okay.
can you conclude it?
Okay, sir.
Okay. Thank you, sir.
So, the impulsion that's generated during the ultrasonication results in the cell lysis or the cell membrane lysis at the surface of the microorganisms, which cause the porosity, which cause the pores, and ultimately affects the microbial damage. So, this ultrasonication is normally used in the transformation or preservation of food and the extraction of various bioactive components from the food system. Then another technique is the packaging, without which the food preservation is not confirmed. As you know, the packaging is used as a containment. It is used to protect and several uh, preserve the food from the several chemical and microbial contamination, water vapor, heat and light, smell, insect, etc. Packaging also used as a means of communication and marketing, that in the form of brand logos, ingredient lists, nutritional facts, etc. It is also provide convenience during the transporting and handling of the food commodity. So, when we are talking about the novel techniques, then in packaging, there are two techniques that are widely spread nowadays used: active packaging and intelligent packaging. In the active packaging mainly use the active initiators that used to preserve the food. Uh, it is introduced in the market of Japan in the mid 70s and raised the attention in the European and American industries in the mid 90s. So active packaging always say that I am active but my work is selective. Why? Because it depends on the which type of food product is going to be treated with the active material. So what type of system is there in the active packaging? It can be used in the form of sachet, pads, and tablets into the packaging material, uh, which can be used as a absorber or the emitter. Uh, Several absorbers such as oxygen absorbers, moisture absorbers, and carbon dioxide absorbers, whereas emitters, it includes carbon dioxide emitters, ethanol emitters, or chlorine dioxide emitters. Whereas active packaging in the form of antimicrobial packaging, it is used uh, as emitters or direct incorporation of antimicrobial agents into the packaging system, or coating the antimicrobial packaging on the surface, antimicrobial polymer, bioactive edible films and coatings, attachment and immobilization of the antimicrobial on the surface of the packaging material. So by direct incorporation of antimicrobial into the food packaging, it is converted it is used in the form of melting foam or solvent compounding, used as a precursor molecules or carriers. It is used as a multi-layer films or used as a nano composite. There is antimicrobial polymers which can be used as a natural and modified, whereas when we immobilize the antimicrobe on the food substrate, or it can be used as an adsorb form on the food surface, polymer surface of the packaging. So how it works? Active, as you can able to see, there is a polymer on which the active layer or the lamination of the active ingredients takes place, followed by headspace and then food. In the second, there is a multi-layer technique in which first protective type layer followed by active layer, followed by one more polymer layer, and then headspace and the food. And the third, you can see the direct covalent attachment to polymer surface. The active ingredient covalently attached
Hello,
to the
Manish
active
sir.
packaging. Hello.
Manish sir, sorry to interrupt you, but sir, please conclude in 2 minutes. It's a request.
Okay, sir. And the fourth one is direct incorporation into the polymer matrix, which may include the encapsulation. So these are some of the products that are available in the market. These are the antimicrobial active packaging, in which the antimicrobial packaging can be used with the bio-based polymers in order to create the polymers in the form of that has the biodegradable properties along with the other properties. So there is a difference between the active and the intelligent packaging. Active packaging mainly used to protect and preserve the food, whereas the intelligent packaging is used to communicate the food, communicate and tells you the history of the food within the system. It contains the active components within it that absorb or release the compounds to the food and significantly show the changes within them. So these are some of the intelligent packaging that include sensors, indicators, tags, barcodes, holograms, and thermochromic inks. Indicators, as we